It's happening right now. Creation Ministries International and Bible Discovery TV Network present the Creation Super Conference of 2011. We're blowing your mind with biblical truth in a stunning display of discoveries in science. Join us now as we go live from Muskoka Bible Camp into the Super Conference with Bible Discovery TV and Creation Ministries International. Good morning, good afternoon, good evening, wherever you are. Rod Hember here. We are live, of course, at Muskoka Bible Center, and this is the Super Conference 2011. Coming up, Dr. Robert Carter, multi-dimensional genome. Now, what in the world is that about? This brilliant scientist who did some amazing presentations, presented last night some amazing things. Now, we were not broadcasting last night, so we're working very hard to get those sessions up on the VOD. They're uploading right now, so we're working very hard to get that established. Thank you to everyone who was praying for us, because we had to go off the air yesterday afternoon, and we had a big problem with the internet, but through prayer and a lot of work, we're back on the air, so praise the Lord for that. Right after lunch, Calvin Smith at 1:30 is going to be talking about dinosaurs, a kind of a question and answer deal going on there. And we're going to broadcast that right here. Now, also, I wanted to remind you before we go inside that we also are on 9330 shortwave radio. I'm very excited about that, by the way, folks. 9330 shortwave radio. If you are a ham radio operator and you want to listen to the conference, let me see if I can find it here. Oh, there it is. I just said that about 20 seconds ago. Anyway, the radio, the shortwave radio, we're broadcasting also Radio 211. So make sure you call a friend, tell a friend, Facebook a friend, YouTube a friend, whatever you do. Get the link, put it in, send it around to as many people as you can, promote it, because coming up is Dr. Robert Carter, renowned, really, what do you call those people? Biogeneticist, something of that nature, some kind of big fancy title. Anyway, he's coming up now, so without any further delay, let's get into the sanctuary and talk about this multi-dimensional should be an echo, Andrew, when you say that. Multi-dimensional genome, I don't know. Anyway, so it's good. And also, briefly before we go, and I want to remind you as well, creation.com. Creation.com. You want to go to creation.com to find all of the resources there. Want to encourage you to consider becoming a partner with CMI, Creation Ministries International, as well. And by the way, just another point I want to make before we go inside. We're waiting for Dr. Carter, and I'm just getting the cue to give a few more announcements. Dr. Danny Faulkner, Universe by Design. Now, we spent some time with him yesterday in the interview room. And Dr. Faulkner is, I'll tell you what, I love the guy. He's awesome. An astronomer who actually is also a biblicist, teaches the Bible. This is exciting to me, because here's a man who knows that the heavens declare the glory of God, and his works, the firmament, display his works. Just previously to this, we had Dr. Mel Silvestro, a geologist, and of course, Isaiah chapter 6 says, the whole earth is filled with his glory. So we're doing all of this, and he's coming up later on tonight at about 6:30. There will be tonight a pre-service worship time, and we'll probably go on the air at about 6:10-ish, something like that. That is Eastern Standard Time, or Daylight Time, I believe we're on Eastern Daylight Time right now. And so that's 6:30 this afternoon. Make sure you tune in early for the worship service. Then Dr. Danny Faulkner, Universe by Design. He's going to be talking about comets and the age of the solar system. Comets and the age of the solar system. And then, as if that's not enough, I feel like the guy selling awnings and Mighty Putty. But wait, there's more. Coming up at 8:30, we'll have Dr. Jonathan Sarfati, by Design. He wants to talk about design, a brilliant scientist, a brilliant theologian. And then later on at 9:45 to 10, right around there, tonight, Eastern Daylight Time, p.m., Richard Fangred, Grand Canyon, A Different View. All of this coming up today. Now, here it is. It's time for Dr. Carter. Let's go inside and listen to this brilliant presentation. Multi-dimensional genome.
Center. All right. I was going to, since Emil didn't do this, I was going to boom for some books up here.
You know I didn't. Because I didn't have time to go back and get them.
Yeah. He mentioned the Ice Age in one of the questions, and mentioned Mike Ord's book, Frozen in Time. It's a great book on the mammoths and how they were frozen, and the buttercups that Emil talked about that are still in their mouths, and so on. So if you're interested in that topic, here's a book for you. Several people have asked, is there a book by Emil that he's done on this topic? Well, the answer is sort of no, but he's a contributor to this one here, Rock Solid Answers. And what this is it's a number of different people, actually, not just Emil, but other geologists, other scientists as well, tackling some of the attempted rebuttals to creationist arguments on sites like Talk Origins and so on. So evolutionists, knowledgeable evolutionists, have come along and attempted to refute some of the creationist claims, and they are defended in this book. A good book on geology at sort of a basic level. This would be good for even for kind of junior high and up, or maybe high school and up. That type of thing is John Morris's book. John Morris is the president of the Institute for Creation Research, the son of Henry Morris. That name might ring a bell. The Young Earth, and that's a great book there on geological things. All right, other announcements. If you're here and you have children, just another reminder that if you see someone making their way to the whiteboard here on the side and writing a number on it, keep an eye on that number, and if that's the number of your child, then you need to go and pick up your child or see what's going on there. It won't be in the lights, it'll be on the whiteboard over here. Has it been on the lights? Okay. Okay, so the problem is the lights only have 2 numbers, and that's the problem. There's a lot more kids here than usual. Speaking of kids and so on, the dinosaur presentation with my colleague Calvin Smith is in here at 1:30. The geology trip tomorrow at 12:30. Note 12:30, not 1:30. We've pulled it back an hour. Emil thinks he needs that time, and he can do more teaching if we pull it back an hour. So tomorrow, if you want to go on the geology field trip, sign up over at registration, and it is at 12:30, not 1:30. And so you need to have a quick lunch tomorrow. This is tomorrow, and then be off with the meal. A number of you have approached us about having speakers in your churches, and what's involved there. And the first question that usually comes up is, how much do you charge? You know what? All CMI offices, all 7 offices around the world, have no set fees. We don't want to hinder small churches from having a meeting like this, to having Dr. Sylvester or Jim Mason or other speakers coming to their church. We don't want to leave those churches out and only go to mega churches. That's why we have no set fees. So it's up to the church to decide. Bigger churches can bless CMI with a bit more. Look, it's anywhere from 300 to 3,000, right? It's the church decides. If you want to have a speaker at your church, we don't want a speaker fee standing in the way of us coming to your church. Okay? So, you can talk to some of us about if you have other questions about getting a speaker in your church. Most of us are from Canada. Our 2011, the fall of 2011, is pretty well booked. Most of 2012 is it's filling up. But weekday meetings, we can always do weekday meetings. You can squeeze them in here and there. So if you want a speaker, think months in advance and so on. And but we can come to your church. It's not a problem. Dr. Robert Carter, are you ready for another session by Dr. Carter? He's a busy guy. What is this, session number 4? It's number 4, isn't it? Yeah, including the night owl session last night. I can't believe how many of you stuck around for the night owl session. Man, you guys, your eyes are going to be popping out of your head by the end of the week. But anyways, Dr. Carter has been introduced a number of times already. Let's just get on with the show. Robert, why don't you come up? All right. Please welcome him.
Thank you. Well, thank you, everybody, for coming once again. You must be gluttons for punishment. If you heard my earlier talk, my first talk, genetics according to Jesus, I tried very hard in that talk to go slow, to define my terms, to make sure we caught up. I gave you the seeker signal to to tell me that I need to stop and help out with another explanation. I'm not going to do that in this talk. I don't have time to begin to discuss the genome, and what it is means we instantly get into territory that's above our heads. Consider this shock and awe. So just hold on to your socks and just hold on for this ride. It's going to be a crazy, wild ride. We're going to talk about the genome. If you're not a subscriber to the Journal of Creation, you are here, therefore you are a creation junkie. Therefore, you should be subscribing to the Journal of Creation. I believe right now that creationists are at the forefront of a revolution in genetics, and we are coming out with some ideas. In fact, the things that I've read in this journal over just the last several years have stunned me, have motivated me, have excited me, and I'm like, wow, what an amazing idea that somebody else has written and I get to read about. Even as a geneticist, I'm learning a lot from what I read from other people. So please look at that. But our discussion right now is on the genome. Well, the genome is made of DNA. And DNA, as you've heard already a couple of times in this week, DNA is the most complex information storage mechanism in the known universe. DNA is a hard drive. The molecule is not important. It's what's written in the molecule. And what's written in the molecule is a very complex code. In fact, the human genome, I showed a different picture of the genome earlier. It's broken up into pieces. You have 6 feet of DNA in every side, every single one of your cells, except your red blood cells, which don't have nuclei. But every single one of your cells has 6 feet of DNA. So right off the bat, somehow God engineered this incredibly skinny. Each one of these, if you have this many, each one of them is probably about this long if you stretch it out. But it's sticky, and it's fragile. And he packed it into something that you can't even see. Could you imagine walking around with 6 feet of string in your pocket? How about 6 feet of thread in your pocket? And you have to pull it out and say, okay, it says go turn left. You have like directions for going somewhere written on the string, maybe a Morse code or something. Okay, now cover it with glue and put it back in your pocket. Now you say, okay, I need the next turn direction. Pull it out. Oh man, oh man, well that's what the genome is like. The genome controls things like biochemistry. And it keeps going. The genome controls things like how proteins interact with one another. This is a map that you're looking at, a map of fruit fly proteins. Each dot on the map is a protein. These are proteins that occur inside the nucleus. These are proteins that occur in the cell cytoplasm. These are proteins that are associated with the membrane or their extracellular proteins. And here we have a zoom up, and here we have another zoom up. Each one of these dots is a protein. This protein, 1, 2, 3, 4, 5, 6. They only showed about a third of all the proteins that they found because you couldn't see anything if they showed them all. In fact, they filtered out all the proteins with lots of interactions with other proteins because all you would see was black lines everywhere. So this is only the lightly interacting proteins. One protein might be shaped like this. To interact with another protein, there's another protein shaped like this that fits on there. Oh, but there's another protein that's shaped like this, and there's another protein that's shaped like this, another protein that's shaped like this. These are specified shape interactions where proteins interact with other proteins in many, many different ways. Now looking at this right here, this is called an interactome. You've heard of a genome, right? It's the sum of your genes. This is the interactome, the sum of the protein interactions in a simple fruit fly. Simple. If you break any one of those links, you're more than likely going to get a deformed or a dead fruit fly. You can tweak the link, make it a little stronger, a little weak, a little bit, but if you break the link, you're in trouble. This is a very delicately balanced system. How did it evolve? Once it's in this delicately balanced position, how does it evolve any further? The human interactome is at least 10 times more complicated. At least. We don't even know how complicated it is because we don't know how many proteins we produce yet. We know we produce well over 100,000 proteins, but that's all we know. And each one of them is slightly different, and they all do different things. So right away, we can conclude the genome is not simple, but it's more than that. The genome isn't static. It doesn't sit in one place. It changes dynamically. In fact, for the rest of this talk, I'm going to base it on this thesis. The human genome is simply the most complex computer operating system in the universe, bar none. But unlike our computer operating systems, the human genome and other genetic information, other species, operates in 4 dimensions. Now, right away you say, what? Watch. 4 dimensions. But first, the computer operating system idea. Here's a paper that appeared last year in Proceedings of National Academy of Sciences. They compared the regulation of transcription. That's when you take DNA and you turn it into RNA. They compared that in the cell of a simple bacterium on the left to the Linux operating system. Right away, you can see there's some comparisons. Right away, you see a lot of differences. In the E. coli, the simple bacteria, there are many master regulators. There are a few master regulators that are controlling many different transcripts of RNA. But in the call graph for Linux, there are a few master regulators very inefficiently calling for the production of a few subroutines. See the difference between this? One of these is designed to be modifiable. One of these is designed to do a complex set of tasks. One of these can be changed. One of these can't. Which one is designed to be modifiable? Which one is easier to change? To change this means you have to change one of these, and to change one of these or these means you're going to affect a whole lot of different functions. E. coli, the organism, the computer operating system in a simple circular chromosome for E. coli is tuned specifically to answer a set of questions in the most possible optimal way. We do not generate computer programs like that because we're not that smart. And our computer programs, like Linux, the operating system, we have to do all these different things. We don't quite know what's going to happen in the future, and we modify it over time. In an evolutionary sort of system, this is an evolutionary system. We can watch the evolution of this computer program over time since 1991 when Linus Torvald invented it. And that's what evolution does. This is what a brilliant designer does. So, I said the human genome operates in 4 dimensions. Let's go through the dimensions. The first dimension of the genome is simply the linear string of letters, right? One dimension is a line. Well, DNA is a line, right? Well, if we look at the first dimension, we get something that looks like this. That's the beginning of the human Y chromosome. That doesn't help me very much. It's just a bunch of letters, A's, T's, G's, and C's. Can you make sense of that? When we sequenced the human genome, we said, aha, we now understand the genome. What do all these letters mean? Well, we need tools to be able to analyze genomic data. You can't just look at letters. First of all, there are too many of them. Second of all, letters aren't intuitively obvious to people. We're very good at pattern recognition. We are really good at pattern recognition. I will prove it. I'm going to replace each one of these letters. I'm going to show you the exact same sequence, but instead of letters, I'm going to replace each letter with a dot. 4 colors. Red, green, black, and blue. 1 for each of the letters. What do you see? Patterns. Boom, there are patterns in the genome. There are patterns all over the genome. There are repeating patterns here and there and up and down. You cannot escape the genome without obviously seeing the patterns. This program, by the way, was written by a very good friend of mine. Y chromosome. Not only that, can you see this pattern, how it shifts a little bit and then shifts a little bit and shifts? This is a repeating element. It repeats 1, 2, 3, 4 times. So there, thousands of times. These are small deletions that set the thing back a little bit. What's this? It's part of the chromosome that we haven't sequenced yet. We can't, in our current technology, sequence highly repetitive pieces of DNA. Because the sequencing machines, when the human genome was made, especially 10 years ago, the sequencing machines generate about 300 base pair long sequences. And so what you do is you sequence billions of 300 base pair things and you line them up. But when you get to a place that has lots of repeats, you don't know where to put that 300 base pair thing inside that repeat. So they left a gap there. Nice, probably 10,000 letters, 100,000 letters, nice round number gap. They said, okay, we don't know this, we'll put a space there, and the letter in the genome is N. So maybe 10,000, 20, 30, 40,000 ends right there. But notice also in the genome, there's some places where different letters appear at high abundance. Different letters. Different letters. Oh, here, look, here's another repeat. But see, I set the window here. This width is exactly 4 times the length of this repeat. Well, this repeat, there's 5 of them in here. It has a different frequency. What is all that stuff? Well, we don't know yet. Now I'm going to do something else. Instead of 1 pixel per letter, let's take 1,000 letters and take their average. So here is the entire chromosome with an average of 1,000 letters at a time. Even though it's kind of murky gray and blue, you can see there's some regions with very different letter compositions. And you can see letter composition different and a gigantic gap. We haven't sequenced half the Y chromosome. Because there's so much repeats in there, we can't do it. Interesting. Okay, so that's the first dimension of the human genome. Everybody understand it so far? We got 3 more to go. Second dimension, you can not draw this in a line. You need like a piece of paper, and you got to draw something here and here and draw a line between them, an arrow, and then an arrow over here and here and here and here. And it's huge meshwork where one piece of DNA is interacting with another piece of DNA in the genome. Sometimes it's a direct interaction, but most often the interaction occurs through DNA or RNA proxies, where this piece of DNA tells this other piece of DNA something using an intermediary. It only occurs in 2 dimensions. This is the 2-dimensional interaction. A lot of us in biology class, we learned about the classic lac operon in bacteria. Well, E. coli, they process sugars. That's how they live. They eat the sugar, they get energy, and they do all their E. coli stuff. But they eat different sugars depending upon what sugar is present, whether or not it's anaerobic or aerobic and things like that. So we have this thing called the lac operon, which is a couple of genes, and they're either turned on or turned off to digest lactose or not digest lactose, depending on the presence of a cap protein or a repressor protein, or the RNA polymerase, and all these different things control the expression of these genes. Well, but this is processed somewhere else, and this is from somewhere else. So this is 2-dimensional interactions. Okay? Well, the second dimension of the genome involves things like this: specificity factors, enhancers, repressors, activators, transcription factors, histone acetylation, DNA methylation, post-transcriptional regulation, where even though the RNA is manufactured, it might not actually turn into a protein, it might do something else or have something else done to it. And then this process called alternate splicing, which I'll explain in a moment. This is crazy complicated. Alternate splicing actually was a giant bombshell that dropped on the evolutionary world a couple of years ago. Okay, that's my next slide. I don't remember how many years ago now, I'm getting lost in time, but several years ago, the ENCODE project produced the results of their study. Now, ENCODE, Encyclopedia of DNA Elements. What they did is they said, we want to look at how much of the genome is active. Because remember, most of the genome is junk, right? 98% of the genome is junk. You've heard that, right? So let's just go look and see how much of this junk is junk. And so they took 1% of the genome. Doesn't sound like much, but that was millions of base pairs of DNA. 1% of the genome. They look at this piece and this piece and this piece and this piece and this piece, a total of 1%. And they were quite stunned because they found out that just about everything was active. Some of it very active, some of it only a little bit active, but just about every piece of DNA in the genome was turned into RNA at some point. That's not supposed to be true. It's supposed to be inactive. But not only that, they found out that most of the genome codes for multiple RNAs simultaneously. In fact, both strands do also. So most of your genomic function is producing RNA, not protein. See, I used to think that we were protein organisms, right? We're made of proteins. The genome codes for protein. The protein makes people. No, we're RNA organisms. We are an RNA computer. Protein is the output. That was huge. This is not supposed to be true. This makes the genome much more complicated. But all these overlapping things, what happens when you have a mutation that occurs in one place? Well, a location in like this red dot here, will affect multiple transcripts. So all of a sudden, Darwinian evolution becomes very complicated. In fact, Darwinian evolution grinds to a halt. It cannot handle polyfunctionality. It needs discrete mutations with a discrete effect, and it has a large effect so the natural selection can find that effect and operate upon it. But if if the average mutation affects more than one trait simultaneously, that means that natural selection can't operate because it can't select for your ability to digest garlic and your heat tolerance and whether or not you like the smell of roses. Random things. Right here, he interacted with one mutation. Okay, let's get into the splicing code. What I have up here on the screen is the double-stranded DNA stretched out, and I'm illustrating in color there a gene. Now, genes are not very simple. They're actually very complicated. Let's get rid of the second strand. Now, in the gene, we've got several interesting parts. First of all, we have the parts that code for protein, the red stuff. They're called exons. And then we have those blue pieces in between. Those are called introns. They don't code for protein. They're actually, when the messenger RNA is made from that piece of DNA, the introns are cut out and thrown away. Some of the introns actually have genes in them. Some of them have other effects in the genome. But just on the base level, they're cut out, they're discarded, and then those red things are joined together to make the messenger RNA for the protein. But these yellow areas in the top and bottom, they don't turn into protein either. These are called the untranslated regions. This is where the DNA polymerase lands. This is where the enhancers and repressors and activators and things bind, and things happen over here also. Actually, sometimes this codes for an independent RNA, so when this is made, a second RNA pops off and does something else in the genome. But let me ask you a question here. I alluded to this in one of my other talks. We've only have about 22,000 protein coding genes in our genome. Those 22,000 protein coding genes manufacture well over 100,000 unique proteins. How do they do that? This is one protein, right? We can only make 22,000 proteins. How can we have so many proteins in our body if we only have that number of genes? Well, the answer was started to be hinted at with the ENCODE project, and since then people have worked out the sequence for it, or beginning to anyway. It's called alternate splicing. It turns out that this exon right here is not just used in this protein, it's used in another protein, another protein, another protein, and another protein. In fact, it turns out that embedded into the end of the exon and the beginning of the intron are, depending on the cell type, depending upon the gene, depending on the exon, and depending on what stage of development, there are between 12 and 19, on average, little teeny signals, little words here and here that tell the cell when to use that exon, under what conditions, in what cell type, under what a time of development. Because you think about it, when you're an embryo developing in your mother's womb, you don't need the same genes you need when you're 80 years old. Likewise, you don't need the same genes you need when you're going through puberty. Likewise, you don't need the same genes as, let's say, I'm allergic to peanuts. So let's say I eat a peanut, right? And my body says, you ate a peanut, get rid of the protein, get rid of the poison. Well, my eyeballs don't need to digest the peanut. My toenails, they're dead. That's a bad example. My base of my hair follicles, they don't need to digest the protein. No, my liver does. So my liver says, I know where that gene is, or I know where those exons for that protein are dispersed throughout the genome. So the liver says, I can do it. The liver finds those exons spread out in the entire genome, combines them together into an enzyme that it then uses to detoxify that protein. So there are instructions there, cell type, and all that. I just said it several times. The instructions are there for actually three-dimensional genome. Because I just mentioned structure. Actually, it's four-dimensional because I mentioned time also, but I won't get into those points. Let's look at the third dimension of the genome. The third dimension is the architecture of the genome. It's the shape. It's a three-dimensional shape of the genome, of the DNA, inside your nucleus. Now, if you look at a standard chromosome, there's a diagram of a chromosome. We have a centromere. We have 2 telomeres at each end. This is very repetitive DNA. Just the same letters again and again and again and again and again and again and again. There's some genes buried in these, but we can't sequence them yet, so we don't know. Generally, we line up a chromosome with a short arm on top and a long arm on the bottom. This is called the P arm, this is the Q arm. That's generally how we see it. Now, if we throw these things into the nucleus, what does it look like? Do they look like this? Here's a cool picture of all the chromosomes, a different color. Are they just thrown in there at random? Well, that's what I was. No one ever said it directly, but that was the assumption behind all the biology that I learned. They're just in there. Actually, no, this is a false picture, because the telomeres are apparently anchored cell membrane This is the nucleus now. I just got a giant nucleus. The telomeres are apparently anchored, at least in yeast. I'm doing centromeres first. There's a place in the nucleus called the nucleolus. This is the place where your ribosomal RNA is made. So the things that manufacture proteins, the RNA is manufactured here, and there's lots of it. So most of the chromosomes have a loop of DNA that comes in here, and each one of those loops codes for the RNA, the RNAs that go into RNA polymerase. So I keep messing that up. Wow, man, I just stumbled over that badly. That was horrible. I totally screwed that up. Okay, forget it. If you look at a chromosome, it looks like, in yeast, I know this is true for the telomeres, but it looks like the chromosome inside the nucleus looks like that, where the telomeres are anchored to the nuclear membrane, the centromeres are associated with the nucleolus, and there's loops of DNA that come through the nucleolus also. So if you look at all the chromosomes, they're actually held in space. So part of the information in the genome is a 3D configuration of the genome. But this is actually way oversimplified. This is actually silly how simplified I just drew that. Okay, here's DNA, double-stranded. Inside the cell, inside the nucleus, it's wrapped around histone proteins. And those are wrapped around each other, and those are wrapped around each other, and wrapped and wrapped and wrapped and wrapped and wrapped, until eventually, you can't see it here, but there's a condensed chromosome that you can actually see under a microscope. But that's not the way it looks in three-dimensional space. It's not this. It's this. Each one of the chromosomes has a specified location in the nucleus. So part of the genomic information is where to put the chromosome. That has an influence of what genes are next to each other, which influences genetic expression. In fact, genes that are used often tend to be on the outside of the chromosome, easily accessible. Genes that are not used often, like maybe the bad peanut detoxification gene, tend to be buried inside the chromosome. Oh, but see, I'm using genes in the wrong fashion. Genes don't exist. Do you realize that? The classic understanding of gene has been blown out of the water since we've discovered alternate splicing. We do not any longer have a definition of the word gene because it's irrelevant. So what if this transcript can be made? The exon is what's important because that's going to be used here, 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 and here. Now, very interestingly, when they started sequencing the human genome, they assumed that when you have a series of proteins that are needed in a row, that the genes for those proteins will be right next to each other in the genome. That makes sense. That's how I would design it if I was an architect. I put everything I needed right next to each other. But when they sequenced the genome, they found out that it was not true at all. And so the first reaction was to throw up their hands and say, see that? The genome's just a bunch of junk. It's just randomly thrown in there, genes here, genes there, doesn't matter, just somehow all works out, until someone examined it in 3 dimensions. And they realized that genes that are used together tend to be clustered together in 3-dimensional space, even if they're on different chromosomes. Oops. So part of that information is built into the 3-dimensional configuration of the genome. Now, I want to give you an illustration of what happens when you ignore this. In my other talk, I talked about the myth of 1% and how humans and chimpanzees, first of all, the number is impossible to calculate because it depends on what you're trying to count. There's different examples of ways to count. But not only that, they're much more different than 1%. Whatever that number is beats me. I don't know how to count it. But when they manufactured the chimpanzee genome, they made a big mistake. I'm showing you a picture here using that Skittle program from earlier. This is human chromosome number 2. You can see human chromosome number 2. It's beautiful. It's nice and dark, meaning there are no gaps in the middle of it, except for there's a gap here at the centromere. There's a couple large gaps inside because we can't quite sequence that. But by and large, this is a wonderfully complete chromosome. Now, I want to show you the 2 comparative chimpanzee chromosomes. First of all, chimpanzee chromosome 2A and 2B. They used to be called, I think, 12 and 13. But they renumbered them because they believed that these 2 things fused together to make human chromosome 2. Long story. That's for another talk. Notice, first of all, this chromosome is upside down. This is the long arm. That's a short arm. It's the only chromosome in any genetic database anywhere that's backwards. They put it backwards, though, because they wanted to be able to line it up here. Notice this chromosome has a gigantic blank space. Why? Because they left it blank, because they wanted to line it up like that. From where you're sitting, Can you say that this is lighter than this? Does this look kind of gray to you? Why? Because there's lots of white pixels in there. There are, in fact, 300,000 gaps in the human genome, in the chimpanzee genome, that don't exist in the human genome. Because when they made the chimpanzee genome, this is back in the dark ages of technology, it cost too much money. It cost us $3 billion to do the human genome. They didn't have $3 billion to do the chimpanzee genome, so they cheaped out. They assumed common ancestry between humans and chimps, never even bothered to test it, and they took the human genome, which was very deeply sequenced, because you have to make lots of copies of these little pieces. I mean, you have to like copy the whole genome 30 times over, and then line up all the little pieces to get the complete genome. They sequenced the chimp genome about 5 or 6 times over, took the human genome, and lined up the pieces on the human genome. And the result was a disaster. First of all, just very simply calculating the size of the chimpanzee genome and the human genome that I have here on my laptop that I downloaded from the government databases, the chimpanzee genome is quite a bit larger than the human genome. But not only that, the human genome has 328,000 blocks, N blocks, spaces, gaps with the letter N in it. And they're usually 10,000 letters long, 20,000, 30,000, nice round numbers. Well, the chimpanzee genome has 318,514 more. Including the big ones, the average size is only 51 letters. In fact, almost twice as much of the chimpanzee genome cannot be sequenced than the human genome because of all these gaps. Plus the fact that telomeres on chimps might be longer, which adds to the unsequenceable parts at the moment. Now, why is this true? This is true because they assume the genome is one-dimensional. They assumed common ancestry. They assumed they could take these pieces and line up beautifully, but when they tried to line up the pieces, they ran into 300,000 places where the chimpanzee pieces could not line up with the human pieces, so they left a gap there. That means that the chimpanzee genome is garbage. It's an embarrassment. It should be thrown out and redone. Cool. You'll never hear that in an evolutionary textbook. Okay, the fourth dimension. Okay, yeah, okay. Everybody okay so far? We just did 3 dimensions in half an hour. You're right? You with me? Okay. Fourth dimension is time. Time. The genome changes in the first dimension. It changes in the second dimension. It changes in the third dimension over time. We have conformational changes during development. As the embryo is turning into a baby, is turning into an adult, different genes need to be expressed, so the chromosomes rearrange themselves to expose different genes. We have conformational changes over minutes, a bad peanut analogy. The chromosomes have to open up, and literally, the nucleus reaches out, grabs a piece of DNA, snips it, unwinds it, pulls it through the other chromosomes to a place where transcription is going to occur, transcribes a bunch of RNA, breaks up all the exons and introns, repackages it to make an RNA that's going to use that protein that I need, puts all the DNA away without ever making knots, and then sends that piece of RNA outside the nucleus to be turned into protein in minutes. There are DNA rearrangements that occur, duplications that occur, and deletions that occur during development. There are some species that have pretty astounding changes in their linear string of DNA during development. Now, I don't know how much or if it happens in humans. I suspect it might, but I don't know. We also have this another time thing. DNA genes can be turned off or turned on, like hard switch, like you're off, and it's really hard to flip it back again. This is called methylation. Your cell can put a methyl group, a carbon with 3 hydrogens, stick it onto a piece of DNA. That means that the transcription machinery can't slide along that piece of DNA. That piece of DNA is turned off. Now, I'm going to use this example. I'm not going to embarrass a person. But there is a person in this room right now who was in her mother, in the person's mother, in her mother, I already said it, during the winter of 1944-1945 in Holland. During this winter, the Nazis were very angry at the Dutch people. They decided they were going to cut off all train shipments, and they tried to starve the Dutch people to death. I have some letters that my grandmother's cousin wrote to her right after the war. Dear Cousin Winnie, thank you for your letter. It's okay. We have a little bit of food because we live on a farm. It's the people in the cities that are having problems. My grandmother's cousin survived the Dutch hunger winter. Many Dutch people did not. Now, interestingly, the women who were pregnant during that time, depending on which stage, which trimester they were in, their babies have had a profoundly influenced genome. The babies that were born after this have come out with a characteristic suite of metabolic disorders. And they're hardwired. And they don't turn off very easily. And it's because the mother was starving. So proteins are a problem, sugars are a problem, all sorts of problems for the baby. Crazy. And some of these things are appearing in the third generation. One reason is because the female baby in the mother's womb has her eggs already finished. So the eggs for the next generation were also starving. This is called epigenetics. And epigenetics is a big problem for Darwinian evolution. Because if you have an environmental effect that affects something 2 or 3 generations later, this person over here might be getting killed off or selected against for something that has nothing to do with it. It makes natural selection really spongy. It makes natural selection not be able to target anything because the biggest effect, epigenetics produces measurable effects. Most mutations don't produce measurable effects. So the measurable effects that natural selection even might be able to operate on, if it operates at all, might be killing off or not killing off people that have nothing to do with actually the mutations they carry. Oh, man. All right, I'm going to share this with you. This has had a profound impact. I believe this one chart has changed my life. This here is, they took a bunch of men who had pre-prostate cancer. They hadn't had prostate cancer, but they had prostate issues. And they took a biopsy. And they said, come back in 6 months. And they took another biopsy. And here, I'm gonna split this chart in half. This is before. This is 6 months later. And it had a control group of men also. And what we're looking at is a bunch of genes. Each column is the genes in a man. And each row is a gene. So these are hundreds, if not thousands, of genes here. That horizontal line breaks the difference between the good genes and the bad genes. Now in this case, green is off, red is on. Bad genes are genes that are dealing with detoxification, fixing things that are broken, stress genes. Good genes are general health genes, building up genes, genes that deal with just general regulation. Look at what happened. Do you see a profound shift in the gene expression in these men? You know what they told the men? Go home, start these general diet and exercise guidelines. Just follow these guidelines. Nothing specific. Just generally, this much exercise, these kind of foods, come back in 6 months, and they had a profound shift in hundreds, if not thousands, of genes that either turned on or turned off just based on a simple environmental change. You see where I'm getting at? The goal in life is to do this. Most people do this. And their life is horrible for decades before they die. We can influence that tremendously just using some simple procedures. Exercise for an older person is a wonder drug. It makes the bone stronger, helps to regulate blood sugar levels, helps to regulate blood fats, on and on and on. Active, well-fed people tend to do much better in life than sedentary, poorly fed people. And what's terrifying is that in our modern world, we are incredibly poorly fed in general because of what we eat. Now, I'm going to take a whole lecture on health and nutrition, but I can't. Here we go. Let me get into another subject. You might have known by now that the genome is actually base 4. There are 4 letters in each bit. At any point in the genome, you can have an A, a T, or G, or a C. We don't build our computers that way. Our computers only can count to 1. It's just literally switches. They're on or off, 0 or 1. So your whole computer only counts to 1, and it starts over again at 0. Well, the DNA, our numbering system, our base 10 numbering system, starts at 0, goes to 9, and then when we run out of digits, we start at 0 again. Computers only count to 1. DNA counts to 4. Why are there 4 DNA letters in each protein? 4 DNA letters in DNA. And why do 3 of those letters code for a single amino acid in the protein? If you take 3 letters, 4 times 4 times 4 is 64. God could have coded for 64 different amino acids, but he didn't. He only codes for 20, plus a stop codon. Why? This is one of the biggest puzzles in the genome. It's called codon degeneracy. Here, what I've listed here are all a codon is the 3-letter sequence that codes for an amino acid. So these are amino acids. There's a stop codon there. These three combinations say stop, stop, or stop. And look, some of them have 6 different codons. Some of them have 1. This is also the start codon, by the way. Every protein starts with methionine, ATG. Why on earth does this system exist? See, degeneracy to an evolutionist, oh, it's just junk. Doesn't matter. You can take this and change it to that, nothing happens. You can take one of these and change it to that. It still codes for the same amino acid. The protein doesn't change at all. Look at all that. Actually, it's not necessarily true. Because, well, first of all, some of these are very common. The transfer RNAs that are floating around in the cell, some of them are very rare. Some are very common. So if you place a very common one with a very rare one, your protein translation might pause as it's waiting. And that actually, in some cases, has been shown to mess up protein folding. And all of a sudden, the protein doesn't work anymore, in some cases. All codons are not alike, necessarily. In fact, after the protein is made, there's a thing called a chaperonin molecule. And I know CMI has a video, and I forgot to put it on my computer beforehand. It's terrible. Maybe we'll show it to you later. After the protein is made, it goes into a chaperonin molecule. And the chaperonin folds the protein up, and it pops out folded. You know what happens if it folds improperly? The protein is thrown away and chopped up. That means the chaperonin molecules put a limit on how much a proteins can change. That means that evolution is constrained by things inside the cell that aren't allowing the cell to change because they know if it changes, it's going to die. Darwin had no idea about that. Now, why are there 64 codons for 20 amino acids? Very simple. Actually, not very simple. Here's the conclusion. To allow for multiple overlapping codes simultaneously. God had to code. When he built the genome, he had to code for protein structure and folding, protein function, based on the amino acid letters in a row, but also the intron-exon boundaries, a little signal at the ends of the introns and exons say, I'm an intron, I'm an exon, cut here. But it says at the same time, it says, oh, put a methionine, a histidine, a guanine, and a cytidine. Oh, sorry, it mixes up, those aren't amino acids. Put these 4 or 5 amino acids there, overwritten at the same time. But also we have all these alternate splicing codes, 12 to 19 little codes right there at the end of the introns and exons, all piled on top of each other. Plus, the DNA is wrapped around histones. Well, the histone protein sticks to DNA that has a characteristic set of nucleotides, a characteristic code. And so within that protein code, there is, at the same time, this letter and this letter and this letter have to be in the right spot so it can fold around the histone. So what this allowed God was to use perfectly acceptable alternate readings in order to code for multiple things instantaneously. It would be like reading this page and realizing that's also a recipe for foie de gras. And it tells you how to fix your outboard motor. Whoa, that's complicated. But that's why we have codon degeneracy. We would not be able to live without it. Now, I want to talk about junk DNA for a second. Junk DNA was a necessary extrapolation from evolutionary mathematics. Back in 1958, Haldane came out with what's now called Haldane's dilemma. He said there has not been enough time in evolutionary history to fix all the differences between humans and chimpanzees. It takes too long. So Kimura in 1968, he said, well, what if most of the genome is non-functional? And then Ono in 1972, he coined the term junk DNA. Most of the genome's just junk, 98% junk, whatever, it just doesn't do anything. Natural selection only has to worry about those little teeny bits that actually do stuff, supposedly solving Haldane's dilemma. Well, interestingly, in the early years of the Human Genome Project, there were calls from the establishment to not sequence the junk DNA. Why would anyone want to do that? Just sequence the coding proteins. We would have missed it. If we had done that, we still don't know how the genome functions necessarily, but we would have absolutely no idea how the genome functions had we not sequenced what they once thought was junk DNA. J.S. Maddock, famous geneticist, he said this a couple years ago. He said, the failure to recognize the full implications of this, particularly the possibility that the junk DNA, the intervening non-coding sequences, maybe transmitting parallel information in the form of RNA molecules, may well go down as one of the biggest mistakes in the history of molecular biology. They assume junk. It's not. So what's the genome? Well, the genome is a multidimensional operating system. It has built-in error-correcting and self-modification codes. And there are multiple overlapping DNA codes, RNA codes, and structural codes. There are DNA genes, there are RNA genes. In fact, most of your genome is designed to produce RNA. And that RNA somehow controls the production of the DNA genes that code for protein. The genome was designed with a huge amount of redundancy on it, on purpose, because without that redundancy, you would already be dead. Because we have redundancy, things can break, and you can have a deletion, and you're still alive because this copy's okay. But if you didn't have that in the first place, you would not be standing here right now. You would all be dead right now. But despite the redundancy, it displays an amazing degree of compactness, because you only have about 20,000 protein-coding genes combinatorially creating well over 100,000, maybe as many as 300,000 distinct proteins. So, everybody understand the genome now? Here's my challenge for the evolutionist. Please, Mr. Evolutionist, please explain to me the origin of the genome. But Darwin famously said, if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Okay, I got to hand it to Darwin. He gave predictable, testable things in his idea, right? So let's test it with the genome. I claim the genome could not have arisen through naturalistic processes. Mr. Evolutionist, I want a workable scenario, including the source of the informational changes, which you don't have, an account of the amount of mutation necessary, which is much too high for you to accept, a description of the selective forces necessary, and selection can hardly find any of the genes, any of the mutations because they're too weak, and you have to do it within a proper time frame. I'll even give you evolutionary time if you like. You cannot do it mathematically. See, Darwin's idea was his grand philosophy. Where he failed was mathematics. That's what Haldane said. That's why they invented junk DNA. Junk DNA has now been thrown out the window. Haldane's dilemma not only has come back, it's come back even worse because the genome's in 4 dimensions, not 1. They don't have the time. The math is not friendly to them. And I'm very excited by that. Now, if you want more info, or if you missed my first talk, it's available on DVD. It's called Mitochondrial Eve and the Three Daughters of Noah. I gave the same talk, essentially the same talk, and we called it Genetics According to Jesus. I had a couple slides in the beginning, and that was all I changed. So it's there if you want to see it again. As you've seen this several times already. Now John Sanford's going to talk about this, I think, tomorrow night. Genetic Entropy and the Mystery of the Genome. Amazing book. It profoundly transformed the way I look at genetics. Just reading this, I cannot tell you how excited I was to read that and realize that just about everything I thought I knew about evolution was wrong. Because, see, the evolutionists, when they find a promising student, they say, come here, boy, let me tell you all the secrets. And when they got someone like me who's slightly a dissenter, they don't tell me the secrets. Sanford knew all the secrets, and he exposed them, and it's an amazing book. I mentioned the Journal of Creation before. Please look at it. Oh, by the way, those are not my fish. Those are the other guy's fish. My fish were better. Oh, well. If you want more, this kind of stuff is we're dealing with design, right? Jonathan Sarfaty's book, By Design, is an amazing, amazing book. It's just page after page after page of really cool facts. If you want to be awed by the designing hand of your creator, that's a great place to turn. You might want to pick up a copy of the DVD, Unlocking the Mysteries of Life, which deals with some of these issues and some cool illustrations, even though it's several years old now, but it's still up to date. It's an amazing DVD. Okay. Well, that was 50 minutes, and I'm very glad to have only gone 50 minutes because I think your heads would have exploded if I added 10 more minutes of information. So, we can open up the floor for questions now. And someone from CMI, I'm sure, will tell me to stop. I guess we have 20 minutes of questions because I went early. Look at that. Okay. Yes, sir. Okay. Okay. He's asking a question about extrapolation. The DNA codes for the production of proteins, but what organizes the proteins into complex structures? Wow, that's a complicated question. It works sort of like this. When you have a single egg cell that's been fertilized, it starts dividing and dividing and dividing and dividing and dividing. And while that's occurring, the genes inside, there are things called Hox genes. These are master organizer genes. When a Hox gene fires, it affects many other genes. And the Hox genes fire in patterns. Pattern, 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 pattern. Which is why, even though my fingertips have never talked to one another, they look exactly the same. Because there's a pattern, like fire, 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 fire. Long distance-wise, the same thing occurred at the same rate in my extremities to produce these. We can see when this fails. People born with different deformities in all sorts of different ways, it's a patterning thing that's misfired. On top of that, we have the egg cell, when it's formed, actually has inherited quite a bit of information from the mother. Not only has it inherited all the components that are inside the cell, but there is a charge there are patterns of charge on the outside of the cell. And when it starts to fold and starts to pinch off, all of a sudden different cells have different charge patterns. And that influences several different things later on. And eventually, as this thing is dividing and dividing and dividing and dividing and dividing, the cells, I would say spontaneously, I know that's not right, but the cells in a certain region of that embryo know that, hey, I'm going to turn into a heart cell. In fact, multiple different cells turn into multiple different parts of the heart. I cannot explain it more than that. I don't know if we know it more than that. I'm sure there are people who are very well skilled in this. That's not my field. All I know that it is amazingly complicated. And so, in other words, if you have a little mutation early on, you can have a drastic effect later on. The hopeful monster theory. Oh, sure, just change the Hox genes around, and you can have a fruit fly born with legs on his fur antenna. Like, yeah, he's going to die. If you change one of these master regulator modules, you create a mutant that is not going to work in real life. They're actually, it's called EvoDevo, evolutionary development, has focused on the Hox genes as the answer to evolution, so they can have a huge change really quickly. But actually, what you do is you destroy a delicately tuned system. Okay. Wow. I skipped that one. Yes? During cellular division, the nuclear membrane, nucleolus dissolve. So what happens to the centromeres and the telomeres? Centromeres line up with one another, and during meiosis, during sexual cell division, the centromeres line up with one another. They cross over like crazy, which means the two arms of the chromosomes are totally independent inheritance units. Because there's so many scramblings, it's random on which one is associated with which one. Somehow they're associated with the spindle. Somehow they're, because when they separate, the centromeres that are pulled apart, and then the chromosomes go like that, and the last thing that separates is the telomeres. But beyond that, I don't really know. Perhaps people are learning that now in high school, and I didn't, but I never really studied it in college either. Great question. Let's Google it later. Sir? There is a very complicated interaction between our genes and our environment to influence lifespan and health. It's very complicated. However, we know the genes that influence longevity. We can double the lifespan of a mouse by tweaking those genes just a little bit. We can also double the lifespan of a mouse by simply giving it half the food it wants to eat. Maintaining a nutritionally balanced diet with half the calories, the mouse will live longer. That's actually been demonstrated in multiple species. It's called caloric restriction. There are certain people groups in the world that live very long. There are people living on the island of Sardinia. It's estimated that 70% of the people on Sardinia are the original inhabitants of Sardinia, or descended from the original inhabitants thousands of years ago. Well, the people living in the highlands, there is an exceptional number of people living to over 100 years old up there. There's a group of people in Japan that are like that, and there's a bunch of little old ladies driving around Loma Linda, California that are over 100 years old also. Those little old ladies are Seventh-day Adventists, and they've been vegetarians all their life. I'm not a vegetarian, by the way. I love meat. But, so here we have genetic things. The people in Sardinia and Japan, genetics. Japan, maybe a little bit of tofu or something. I'm allergic to soy, so I don't eat it. But maybe something in diet. But those little old ladies, it's diet. It's lifestyle. Now, they're not out there running marathons, right? They're not, lifting weights like Arnold Schwarzenegger. That's not how to live a long time. The way to live a long time, keep your brain active. Keep your legs active. You know when people die? It's when they sit. And when a person breaks their hip when they get elderly, oh man, it's very often a downhill trip that's very quick. It affects your heart. It affects everything. So keep your legs strong. So there's a very complex interplay between genes and environment that influence lifespan. We've now figured out, they've done a lot of genetic studies on very old people, and they figured out that there are maybe 10 or so things that they've tracked that influence longevity. And they said, wow, as long as, it doesn't matter which ones you have, but if you have enough of these longevity factors, you'll probably live to be very old, even though you carry the same things that will kill all of us off earlier. So they're longevity genes, and we know they exist, and they're there. And so the next question, can we live to 200 or 300 years old using modern technology? Probably, but I don't know if I want to live that long anyway, because I can't guarantee that modern technology is going to be able to keep my brain alive. I can't guarantee that some other effect's not going to happen. I don't want to live to be 300 years old if I'm decrepit. I don't mind living to 80 or 90 years old that way. God willing, I wouldn't complain too much. Might hurt a little bit, but I don't want to artificially prolong my lifespan if life's not out there. I'm not giving an end-of-life argument, by the way. I'm trying to avoid that. So I just traipsed into that territory by accident. Sir? The question is, do I or anyone else I know what Francis Collins' response is to any of this stuff? I don't suppose you were here for my talk last night, my late night talk? Okay, I talked about Francis Collins. And I said, Mr. Collins, there's abundant evidence that Adam and Eve really did live, and I'm using your own data, and you're saying they didn't live. I don't know what his response is because I haven't heard a response from anything we've written from them. I just know they're ignoring what we write. And I'm trying to change that. I don't want them to ignore it. I want them to acknowledge it because I don't think they're correct. Well, by giving a video that's going to be online, by writing articles, by talking to people about evidences for creation. Eventually, with 7 degrees of separation or whatever. In fact, there's even less than that. I bet there's 2 degrees of separation between me and Francis Collins. Somehow. Maybe 3, the most. Sir? What simple cell? You mean bacteria in evolutionary terms, or is all this complexity I just talked about stored in the simple cell? It is stored in the cell, but what we have learned is cells are absolutely not simple. In fact, Darwin needed the cells to be simple. To Darwin, inside the cell is just a bag of salts. It's protoplasm. Actually, the word protoplasm has an evolutionary connotation to it. But just protoplasm inside the cell. And so for him, it was easy to imagine a cell changing shape or color, whatever. But once we learn the mechanism inside it, evolution is becoming less and less tractable every day because of complexity. Complexity is the antithesis of what evolutionary theory needs. What does Francis Collins say about the number of genes? Well, he was the head of the Human Genome Project, so he pretty much discovered the number that I shared. What? Well, the latest estimate depends. There's arguing and bickering, but the numbers you see are about 22,000, sometimes as high as 25,000 genes. How many cells in the human body? There are several trillion cells in the human body. And there are 3 billion nucleotides in the genome, but you have 2 copies of your genome. How do you get those 3 billion nucleotides to generate 100 trillion or whatever cells? It's in the genome. The information's in the genome. I don't know how it works, sir. Oh, yes, it's compressed. There's compressed, zipped information in the genome. In fact, I have an article in, I think it's the latest Journal of Creation, where I asked the question, can mutation produce information? Well, I say yes. If you define mutation as genetic change, God could have written into the genome a lot of hidden information where a recombination event or a jumping gene or something can reveal information that was in zipped, compressed form. But it's a long argument. It's actually a long paper. I'm going to go back there in a second. Sir? When they come up with the idea of junk DNA, did they come up with a way to say that, a way for lots of mutations to occur in a junk DNA and only a few mutations to occur in the genes that are necessary to sustain life? No, they just had mutation random. As long as the target was small, the likelihood of getting mutation in the good stuff was small. But they also thought that their number of mutations to them was small. Actually, Haldane also said that if there was 1 mutation for every 10 people, we'll go extinct. Because that means that 10% of the population would have to be killed off to get rid of the mutations. Well, there's 100 mutations per person. He's 3 orders of magnitude off from the extinction level. So we're 1,000 times more driven to extinction in his maximum sustainable level that he picked in the 50s. So no. Okay, somewhere back there. Yes? What does DNA and RNA stand for? DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Sorry, I'm trying to do 2 things at once. I don't have a great picture of DNA, at least not a skeletal picture, but I have this space-filling picture of DNA. Right there. That's a picture of DNA. There are 2 chains. There's 1 chain here, and there's another chain here, and they're wrapped around each other. And these things aren't actually attached to one another. They stick together because of positive and negative charges. They're like little magnets. You can actually take these and pull the 2 strings apart. At least a cell can do it. So that is DNA right there. And these red things, these are A's, T's, G's, and C's. A, T, G, C, C, G, T, A, bonded together across the 2 chains and linking the chains together. RNA doesn't do this. RNA doesn't generally form a double helix. RNA is a single-stranded molecule that's extremely fragile. See, the double-strandedness of the DNA, plus a little bit of the chemical differences, help it stay together and help it from destroying itself. RNA breaks down very rapidly. Sir? How often is the DNA replicating itself? Well, every time one of your cells divide, the DNA has to replicate, and that's going to probably happen about 1 million times a minute in your body. I don't know, 1 million times? If you think about it, probably that many. I don't know. I forget how fast it occurs. You can watch it in real time. I don't remember how many minutes the cell takes to replicate the entire 6 billion letters in your genome, but it's incredibly fast. Faster, certainly, than we can read. Yes, way in the back. John Sanford, are you going to talk about human chromosome 2 in your talk? No. Okay. There's an evolutionary argument. In fact, I think if I show this one right here. Many years ago, when people were doing karyotypes of humans, they also did a karyotype on a chimpanzee. And they said, look at that, chimpanzees have less chromosomes than we do. They were missing a pair of chromosomes. Where is that pair of chromosomes? Well, they think they found it here. Our chromosome number 2. Chimpanzees, I think it was 11 and 12. If you look at chimpanzee 11 and 12, the banding pattern here and here matched up with this part and this part of chromosome 2. They said, aha! In evolutionary history, humanity had a chromosomal fusion. And our chromosome 2 is a result of the fusion of 2 ancestral chimpanzee chromosomes. Now, they said, this is a confirmation of evolutionary predictions. Actually, it was no confirmation of anything whatsoever. For the following reasons. First of all, looking at the data of human chromosome 2, there are problems in saying, oh yes, this is a fusion event. Basically, they say that these 2 chimpanzee chromosomes did this and fused, but the telomeres are missing. And the second centromere has been degraded to the point of almost unrecognizability. So when did this occur? How did it occur? We don't know. Also, it wasn't actually a prediction of anything for this reason. Since we have different numbers of chromosomes, they said, aha, look at that, proof of evolution. But if we had the same exact number of set of chromosomes, they would have said, aha, look at that, proof of evolution. Heads I win, tails you lose. So it didn't matter what the answer was, they were going to win the way they set up the argument. Now, I do believe that I don't necessarily believe that there was no chromosomal fusion in human history. I don't believe there's 2 chimpanzee-like chromosomes or 2 ancestral chromosomes fused, but it might have been 2 human chromosomes. I believe we need to study this more. And the reason is this. We have seen chromosomal changes in species we know about, the difference between horses and donkeys. Chromosome count. Does everyone in here believe horses and donkeys are the same created kind and there's only 2 horse-like critters on the ark? Well, that means that in those last 4,000 years, a chromosomal change has occurred in this lineage to produce viable species that hardly can interfertilize anymore. They make a mule, only a very rare mule is fertile. There are like 13 species, excuse me, Australians, I might mess it up, but there are like 13 species of rock wallaby in Australia. They can be all differentiated based on their chromosome count, their karyotype. The house mouse and common mice species, they have a credible array of different chromosome counts. So chromosomal changes can occur. I don't necessarily believe it has occurred. I think it needs to be tested. I think the evolutionists just waving a big wand over and say, look at this evidence of evolution. But there's significant things in human chromosome 2 that should be true and are not, plus the location of these things, and plus we haven't studied fusion and breakage events in other species really to know what we're talking about. But there might have been a fusion in human history. I'm just leaving that door open, just to dot my I's and cross my T's, just in case. Fair enough? Okay. All right. I restarted my clock. I don't have any time. What time is it? 2 minutes. Okay, 2 minutes. Sir?
You mentioned peanut allergies.
Yes.
Food allergies in general, is it that the protein in the food being misidentified, or is it that the liver itself is turned off that part to be able to process it?
I mentioned food allergies. Is it that the protein is being misidentified, or is it the liver just turned off to process that protein? Honestly, I have no idea. All I know is from trial and error, I can't eat corn or peanuts or soy and have to minimize wheat. When my wife took me off of soy, I lost 15 pounds. So I'm back again. I have no idea. Just me. Sir? Is there something called the human genome meltdown? John Sanford is going to talk about genetic entropy. Hey, John, are you going to talk about genetic meltdown? Okay. Yes, there's such a thing called genetic meltdown. It's basically once you accumulate too many mutations, you get a runaway mutation process that will rapidly lead to extinction. It's actually in the evolutionary theory. They worry about it. They don't know how organisms that are clonal, they just have mother's daughter, mother, daughter, mother, daughter. They don't have any men in there. They don't know how they live over time because of the ratcheting mechanism of one mutation after another, and that clonal line should drive them extinct. Interesting stuff. Time, okay. Thank you all very much. Have a good lunch.
Probably one of the best presentations that I have heard, and that is Dr. Robert Carter. The multidimensional genome. What an amazing presentation here at the Super Conference 2011, Muskoka Bible Center on Bible Discovery TV and Radio211.com, 9.330 megahertz around the world on WBCQ, broadcasting live from southern Ontario near Huntsville. Coming up in just a few moments, we're going to be, actually more than a few moments, in about an hour and some time, we're going to be listening to a discussion from Calvin Smith related to dinosaurs and the Bible. Now, if you believe that dinosaurs existed long before man, then you are in evolutionary thinking and under an evolutionary spell. We're going to be talking about that and more with Calvin Smith when we return this afternoon. For now, we're going to return to our regularly scheduled programming, both on Radio 211 and BibleDiscoveryTV.com, but be sure and join us a little bit before 1:30 p.m. this afternoon when we'll be talking about dinosaurs in the Bible. I'm Rod Hembry. Thank you for joining us. We'll be back in about an hour and 15 minutes. Stay there.